the screen is visible. Okay, so we'll be solving the questions today related to amino acids, how they make the functional protein, their polypeptides, and how to predict their structures. Okay, let's begin the session for today. So this is the first question. This has been asked, match the amino acids with their properties. In one column, the amino acids one word coding is given, and another the properties have given. So you have to match it correctly. So before matching this, let's first very briefly understand. So amino acids, we have to see how many categories. Okay, please guys try to join on time. One by one people are joining. Okay, so if I have to divide amino acids, okay, I can divide it in three categories. Amino acids, I can divide into three categories. One is hydrophobic, another hydrophilic, and aromatic. So very easily we can divide it into these three categories. Okay, so I have, so in hydrophilic category also we can divide it into furthermore. So in hydrophobic we have the uncharged or aliphatic amino acids. So very simple technique, gav limp. Okay, only this if you can remember, gav limp. Okay, glycine, alanine, valine, leucine, isoleucine, methionine, and proline. Okay, these comes under the hydrophobic categories. Okay, in the aromatic we have WFY, tryptophan, phenylalanine, and tyrosine. And in the hydrophilic also we have two divisions, one which is polar uncharged, another polar charged. Okay, polar uncharged, another polar charged. So in polar uncharged is STNQC, and polar charged we have further positively charged and negatively charged. This is HRKDE. Very easy to remember, GAV limp, WFY, STNQC, and HRKDE. This is the very simplified classification if you want to do. Okay, so here we can start with the glycine. Okay, so glycine is the simplest amino acid. Okay, so in amino acid we have a center carbon. Along with that we have a carboxy group, carboxylic acid group attached, and this amino group attached, one hydrogen, and here we have the side chain. Okay, so it could be anything, so that is why it is basically referred as R. Okay, it is R group. Whatever it is being added here, according to that we have different amino acids. So in case of glycine, in place of R we have H. Okay, so we have 20 standard amino acids, right? Out of that only 19 are chiral, and this one amino acid is achiral, and that one amino acid is glycine. Okay, because for chiral carbon we need to have all different 4 different moieties need to be attached to the center carbon. If all the moieties are different, then this carbon is known as chiral carbon. But in case of glycine, 2 hydrogens are attached, so that is why it becomes achiral in nature. So glycine is the only amino acid which is achiral in nature, and because of that simplified amino acid, because of that it is the simplest amino acid, and it can attain any kind of conformation. So it is generally found in the turns, loops, in the helices, loops. So this is generally present there. Okay, and due to that reason glycine has the largest area in the Ramachandran plot. So it is the simplest amino acid and only achiral amino acid in the category of amino acids. Okay, so in place and if in place of this glycine, if we have something else, for example in place of R we have this methyl group associated, then it becomes alanine. Okay, so here you can see all different 4 different moieties attached to this particular carbon, and this that's why this is a chiral carbon. So this is the C alpha carbon. Okay, this particular carbon is known as C alpha carbon, and here after that whatever chain will come, it will be having different nomenclature. So this carbon is alpha carbon, this carbon is beta carbon, and further if more carbon, then it is gamma, delta, epsilon, different nomenclature. So this is alpha carbon. Okay, after that, okay, so we have glycine, alanine, then GAV, we have valine. Okay, valine, leucine, isoleucine, if I'll draw. So this is the basic backbone I'm making. So we have something like this, valine. Here CH3, CH3, CH3 at these 3 points. Okay, and then again we have isoleucine, sorry, leucine. So leucine we have a structure like this. Okay, this is leucine. Okay, and then we have isoleucine. So the isoleucine structure is something like this. So this is where my alpha carbon is there, and then I have my another carbon. Okay, okay, so this is beta carbon and that is alpha carbon. So in other amino acid this alpha carbon is the chiral carbon. Generally this alpha carbon is the only chiral carbon, but isoleucine is the only amino acid where you can see 2 chiral carbon, this particular one and also this particular carbon. Here also you can see 4 different moieties attached to the this beta carbon. So this is why that this one also becomes the chiral carbon. So there are 2 chiral carbon molecules present in one amino acid, that is isoleucine. Okay. After that, if we have methionine, so methionine is the only sulfur contain not only, so methionine is one of the sulfur containing amino acid, we know. So we have covered glycine, alanine, valine, leucine, isoleucine. Methionine is the sulfur containing, and then we have proline in this group. So just remember Gav, limp. So there we have proline. So proline is such amino acid that its beta carbon is involving the nitrogen and making a pentagon ring here. So this is a proline structure. Okay. So the side chain of proline is directly attached with the alpha amino group. So due to that, rotation of side chain becomes impossible, and due to this rigidous nature, proline is never found in alpha helix or beta sheet. If it is present, it is involved in making loops or kinks. Okay. So we have covered all these hydrophobic amino acid. Now we have aromatic amino acids. Okay. So we have tryptophan, phenylalanine and tyrosine. Okay. So yeah. So we have these aromatic amino acid. So in aromatic amino acid, what happens? This is my structure. This there is a benzene ring attached to that. Okay. So this is my phenylalanine. Then we have tyrosine, and we have tryptophan. This is NH. Okay. So we have phenylalanine, we have tyrosine, and we have tryptophan. Okay. So we have these aromatic amino acids. So the aromatic uh, amino acid absorb the maximum light of 280 nanometer wavelength. Among all the different amino acid, these amino acid absorb the maximum light. So for detection of protein in the sample, these are the amino acid which are responsible for the detection of protein in any sample, because you can simply take the absorption at 280 nanometer, and if these amino acids are present, it will give you a strong absorption, and that will tell you the concentration of the protein. Okay. So at 280 nanometer, it absorbs most of the light, and that absorption happens because these aromatic acid have conjugated bond system in that. Double bond, single bond, double bond, single bond. That is the conjugated electron system that is responsible for the absorbance of the UV region light. Okay. So if we'll see the order of absorption, so if this is the absorption, the least absorption of light is found in phenylalanine. More than that, we have tyrosine, and tryptophan absorbs maximum light because tryptophan has 2 such aromatic rings. Okay. And here, compared to phenylalanine and tyrosine, the OH is also involved in this conjugated bond formation. So that is why this particular tyrosine absorbs more light than the phenylalanine. Okay. So that is how it has been utilized in the protein estimation. Okay, after that we have another this hydrophilic amino acids. Okay, so in hydrophilic also we have polar but uncharged. We have polar uncharged. We have polar uncharged amino acid. So where there we have I have S, T, N, Q, and C. Okay, so we have serine, threonine, asparagine, glutamine, and cysteine. So these are the amino acid which are polar uncharged amino acid. Okay, and we have polar charged. There we have acid positively charged and negatively charged. We have that histidine, lysine, and arginine. Okay, sorry, it is lysine. Yeah, so these three. So here also we have and here also we have two acidic amino acids which are aspartate and glutamate. We have aspartate and glutamate. So these are the amino acids which are charged generally, and these are the found in the outer surface of the protein because these are involved in making the interaction in any solvent. So that is why these are generally present in the outer surface of any globular protein. Okay, so okay, and also in case of proline, so yeah, in case of proline also one thing. So here generally in case of protein structure, so we have this NH2. So this is the primary amine which is NH2. Okay, if it is a secondary amine, okay, it becomes if it is tertiary amine, one more is replaced. Like this is the primary amine, secondary amine, tertiary amine. In case of secondary amine structure, this is called the imino group. Okay, imino group. So in case of proline structure, we can see that this particular uh, nitrogen is the secondary amine structure. So that is why this particular ring in proline is referred as imino ring. Okay, which is showing or giving the rigidity to this particular amino acid. So back to the question. Now we have these amino acid with us, and this is the properties with us. Now we have to match it according to their properties. So the first is WYF. Okay, so in our list we have this WFY. So it is lying under which category? The first one will be matched with which of the option? A, B, C, D, E. Which one it will match? Please answer. It comes under which category? Just now I told that it comes under the aromatic amino acid categories. So these three amino acid will be coming under the aromatic amino acid categories. Next we have P, which is proline. So out of other remaining option, proline will be matched with which one? Please unmute yourself and try to interact. Try to answer.
Immunogroup.
Immunogroup. Yes. So proline will be interacted with or matched with immunogroup. Yes. So we have first with C, second with E, and third will be, it is IBAL. So these are like isoleucine, valine, alanine, and leucine. So these are all aliphatic amino acids. So out of the options, we'll have this will be matched with this D.
A option.
No, these are all be matched with amino acid with aliphatic side chain. Okay. Now we have this fourth, which is H, H is
Neutral.
H is neutral. Why is it neutral? H is histidine, right? So histidine is a positively charged amino acid, right? H is histidine, which is a positively charged amino acid. So why it will be neutral? So histidine has a pK value between 6 to 7, near this region only. Okay. So what is pK value? Anybody know? So pK is the it denotes the acidity or basic nature of any particular component, right? So the lesser the pK, the more acidic the amino acid will be. Okay. So all the acidic amino acid or all the negatively charged amino acid have very low pK value. However, the basic amino acid or the positively charged amino acid have a higher pK value. But there comes the exception, which is histidine, which is a basic and positively charged amino acid. So histidine, lysine, and arginine. So these two have pK value very high, in between 12 to 13 some range. Okay. But this histidine has the pK value near 6 or 7. Okay. So it has such a low pK value, and because of that, when this particular amino acid present at the neutral pH or physiological pH, it function as a neutral amino acid. Because what happens, pKa is nothing but a pH where there is an equilibrium in the charge or in the two forms of amino acid. So this amino acid also have two forms, protonated and deprotonated. Okay. So this histidine also have a form which is protonated. Okay. Protonated form and deprotonated form. So at pK, both forms are present in the uh, equivalent or in equilibrium. So but when you goes above the pKa, or a pKa is denoted with the at the pH at which the both the forms will be in equivalent manner. Okay, so when you goes above the pKa, you will get, or the deprotonation happens. Okay, and at lower pH the protonation happens. Okay, so at lower pH you have much more H plus ions. Okay, so where it is very easily to get protonated at lower pH, and at above pH the deprotonation is happening. Okay, so as I told that this histidine has a pK value of 6, so when it goes above that, which is 7, that is the physiological pH, the a particular amino acid will undergo deprotonation. Okay, so it will undergo deprotonation, and because of that it will present in its neutral form. Okay, so in histidine structure we have something like this: CH2 in this particular ring, and this is how it looks. Okay, but at physiological pH, which is above its pKa value, the deprotonation of this particular amino acid will take place. So because of that it will now have neutral charge on it. Okay, it will now have neutral charge on it. So that is why at physiological, so here the peak here below the pK it will be in positive charge state, but at above the pK or at the physiological pH it will be having zero charge. So because of that here the fourth option will be matched with first A, that is neutral at physiological pH. Okay, then we have fifth one, which is D and E, which are, we already know that is the negatively charged amino acid or acidic amino acid. So the correct option here would be, so one is matched with C, second it's matched with E, third is the amino acid with the aliphatic side chain, it's D, fourth we are matching with A, and fifth will be matched with B. Okay, so this will be the correct option for this question. Okay, let's move ahead. Okay, so here the question is asked, is dash is also known as Edman's reagent. So Edman's reagent, why is it used, Edman's reagent? Edman's reagent, for what purpose it is used? Anyone? To find out the sequence of the protein, right? Like you have a protein or a polypeptide and you want to know what are the amino acids which are forming this polypeptide. So you want to know the sequence of the polypeptide. For that you use this Edman's reagent for Edman's degradation. Okay, other than that we also have another reagent, which is via N-terminal analysis you get to know what are the amino acids. So this FDNB, which is uh, 1-fluoro-2,4-dinitrobenzene, FDNB, and densyl chloride, they both are not Edman's reagent, but they come under the Sanger's reagent category. That uses N-terminal analysis to find out the which is the amino acid is present at the N-terminal. But for the proper sequencing of each and every amino acid, we generally use this phenyl isothiocyanate. Okay, so here also, suppose you have a chain of amino acids, one polypeptide, second, third, okay, with both everything joined together with the polypeptide bond. So you have this phenyl isothiocyanate, which is a ring structure. Okay. Okay. So this particular thing will make a bond with the amino terminal of the protein. We have one amino acid. This is the peptide bond. This particular thing is the peptide bond, followed by another peptide bond. Okay. So here, and this phenyl isothiocyanate reacts with the first one, make a label it or make a duct, and then further will be cleaved by trifluoroacetic acid activity, and this particular one will come out with this particular amino acid, leaving the second amino acid as it is, and this one will come out. So based on that, this particular one will get sequenced or screened for which amino acid it is, and like that, one by one it will be labeled, and all the amino acids will be sequenced, and that will tell the order of the amino acid in the polypeptide sequence. So Edmonds reagent is phenyl isothiocyanate. Okay, it is a aromatic compound. Okay. Next we have dithiotrietol, DDT, and beta mercaptoethanol is used for what? In peptide sequencing. Okay, so these DTT and beta mercaptoethanol everybody must have heard. For what purpose it has been used? Suppose you have a protein structure and or a protein.
Reducing agent,
Yeah, these are reducing agents, and which bond it will break?
disulfide.
Yes, so these are the reducing agent which will break the disulfide bonds present in the amino acids. Okay, so disulfide bonds are generally formed between two cysteine residues. So cysteines are the sulfur containing amino acids. Okay, so this is one sulfur joined. Then we have like this, we have disulfide bonds present. So these are the disulfide bonds, and after disulfide bond, this disulfide, it is referred as cysteine. Okay, so the reducing agents which are used, that will break this disulfide bond, and it will look something like this. Okay. So that's how it work as a reducing agent, because it incorporates the hydrogen, and incorporation of hydrogen is the reduction of any molecule. So by reducing it, it breaks the disulfide bond, adds the hydrogen to it, and that is why it is referred as reducing agent. Okay, so if after the reaction it is performing reduction, it is referred as reducing agent, and if it is performing oxidation, it will referred as oxidizing agent. But in case of DTT and beta mercaptoethanol, they reduce the cysteine, the sulfur present in the cysteine structure, and that is why work as a reducing agent and breaks the disulfide bond. Okay, so next we have secondary structure of protein is stabilized by. So we have four options. So hydrogen bonding of R groups of amino acid, disulfide bridges between cysteine residues, hydrogen bonding between CO and NH of peptide, hydrophobic interaction of side chain. So secondary structure. So simply, if we have a chain of amino acid joined by polypeptide with each other, a linear chain, that is the primary structure. Then we have the interaction among the chain or among the polypeptide. There comes the secondary structure. So it has been asked, in the secondary structure, how it is associated, okay, how the bonds are formed. So okay, so first is hydrogen bonding of R groups of amino acid. So secondary structure, in secondary structure, which kind of bonding we'll see? Hydrophobic, electrostatic, hydrogen bonding. Which bonding is present? There are several types of bonds, right? Hydrogen bonds, okay, hydrophobic interactions, electrostatic interactions, disulfide bridges. So which bond it is present in the secondary structure? What are the secondary structure of amino acids or polypeptides? What are they called? Alpha helix, beta sheet. You must have heard that, right or not? I will say electrostatic interaction. Correct me. Which bonding is present? Unmute yourself. Try to interact. Please answer. This is very easy. I know everybody knows that. Please answer.
Hydrogen bond between amino acids.
Yes, hydrogen bonding between amino acid. So here our two options are automatically wrong. So fourth option, which is hydrophobic interaction, that is wrong, and we have disulfide bridges, that is also wrong. Okay, these are present in the tertiary structures, these bonding. Now we are left with two option of hydrogen bonding. One is saying between the R groups of amino acid, another is saying between the CO and NH of peptide bond. Which one it is? Sorry, sorry. So we have the hydrogen bonding. So
R group of amino acids.
no. So here you need to understand what is hydrogen bonding. So hydrogen bonding is a type of, like, a weak, comes under weak interaction category. So hydrogen bond takes place between the two electronegative atoms, okay, or and so suppose we have oxygen and nitrogen. So electronegativity is these three. Okay, fluorine, oxygen, nitrogen. So this is order of electronegativity. So fluorine is highly electronegative, then oxygen, and then nitrogen. Okay, so the two atoms should be electronegative, and then we the hydrogen will be attached to any of the uh, any of the electronegative atom, and then there becomes one bonding between hydrogen and other electronegative atom. Okay, so this particular bonding is referred as hydrogen bond. Okay, it could be like opposite also. Okay, like this. Like this. Okay, like this. So this is the hydrogen bonding definition, and the more the electronegative will be the atom, the more stronger will be the hydrogen bond. Okay, so this is first, second, third, and fourth. This one oxygen having more electronegativity than nitrogen, and both the atoms which are involved in the fourth is the oxygen. So this is the strongest interaction, fourth. And then we have, yeah, so then one more thing comes, like which on which one it is associated. Like hydrogen is associated with more electronegative, so here we can see hybrid in the first two, but hydrogen is associated with more electronegative, then this one is comes later for the like weakness, and then we will be having second, and then we'll be having third. So this will be the order of the hydrogen bonding strength. Okay, so in case of amino acid structure, so we have here so we have this alpha carbon, then we have CO NH2 CONH, then we'll have another carbon. So this is, if this is N, this is another carbon, N plus 1. This is alpha helix I am drawing here, and the R chain, it will be associated with the R chain. Okay, and we have again, okay, again C alpha, which will be N plus 2, and we have such, and then we'll come to the N plus 4. Okay, okay, CONH, and then we have this N plus 4. Okay, so then the hydrogen bond forms between the first this nth carbon, and then the fourth carbon, N plus 4 carbon. So there the hydrogen bonding happens. So here the hydrogen bonding happens in such a way, like this. Okay, so it happens between this CO and NH groups. So here the R groups are not involved, because it is for making hydrogen bondings you need an electronegative atom, and that is not necessarily present in the R group. If it is happening, that comes under the tertiary structure bonding, but the secondary structure are generally formed between these kind of CO and NH group, because here you can see oxygen is electronegative, then we have nitrogen, which is electronegative, and along with the nitrogen we have the hydrogen associated with that. So this oxygen and this hydrogen will make the bonding, that will be the hydrogen bonding. So in this particular option we'll be having correct option will be hydrogen bonding between CO and NH of the peptide bond, not this one. Okay, let's move ahead. So now we have intra-chain and interchain disulfide bonds are present in this and this respectively. So it has been asked for disulfide bond. Okay, so we have like we have also intra-chain and interchain hydrogen bonding also, which is present in the secondary structure. For example, just now here this is in the same polypeptide chain hydrogen bonding is happening, so this is intra-chain. And if there are two different chains connected with each other with the hydrogen bonding, that is interchain. So if it has been asked in sense of quaternary tertiary structure, so intra-chain, like there is a polypeptide, okay, then it is making some based on the structure it is forming helix, okay, loops, then sheets, okay, helix. So if this is the structure which is being formed, okay, this is the secondary structure. Then in between this chain only, if disulfide bonds are formed, that is making a compact protein, okay, making it more compact, okay, something will be three-dimensional, I cannot draw it, okay, so something compact which is happening inside, okay, the disulfide bonds which are being formed in the same chain to give the conformation of the protein. So that is called the tertiary structure of the protein, okay, where hydrophobic interaction, disulfide interaction, and all the different kind of interaction is happening in the same polypeptide. So that is the tertiary structure. And if it is interchain, interchain means this is one polypeptide and there will be another protein, okay, another globular protein, and these two proteins are being joined by disulfide bridges, that is making the quaternary structure of the protein, okay. So intrachain, so intrachain will be found, intrachain disulfide bond will be found in the tertiary structure, and interchain disulfide bond will be formed in the quaternary structure, okay, so this will be the correct option. Yeah, so which amino acid is branched at beta carbon, okay, so which one is branched at beta carbon, please answer. Branched at beta carbon, or you can associate it with the chirality also. I just now told that we have this particular carbon which is C alpha, and then it has been asked beta, so it is being asked about the branching, the beta carbon is branched, okay. Out of these four, which amino acid is branched? We just now discussed one, achiral amino acid. Which one is the achiral amino acid? Sorry, not achiral, amino acid which has two chiral centers, sorry, yeah. So this alpha carbon we already know, except glycine all the amino acid are chiral in nature, and this particular amino acid has two chiral center in its structure, okay, that is what the branched is asked. So which amino acid is having dual chiral center in their structure?
Isoleucine.
Yes, it will be isoleucine, okay, we have isoleucine. Something, something wrong with this, but yeah. Now we have like what cannot be determined by using circular dichroism spectroscopy. So why we use circular dichroism? For what purpose it has been used? Circular dichroism. Why is it used? So circular dichroism spectroscopy is used to predict or to find out the secondary structure of a protein. Okay, because this graph you can see, so CD spectra will give you a graph something like this. Okay, something like this. So here alpha sheet will come in this particular dimension only, and then we have this beta sheet which will come in the negative uh, sign in this particular dimension, and then we have the random coils, this green one which will be the random coil. So this is the ideal, or this is the where the positioning of alpha helix, beta sheet and random coil. So if you are running a protein and then you are observing a graph like this only, okay, so that means it will predict that your protein has alpha helix in its structure. Okay, and otherwise if it is having beta sheet in its structure, so that it will predict. So but it will only be used to predict the secondary structure of the protein. So first one is correct. Then protein folding and unfolding as a function of temperature. So same protein, you as a function of temperature, you uh, run both the sample. Okay, one treat with your different temperature, and then you can compare the folding whether there is some because one graph, one untreated will give, and some another temperature you will get another graph which will like which now will having the change peaks. So that will on as the function of temperature you can say the folding and unfolding. Also the conformational changes due to protein protein interaction. If only single protein is given, you are getting alpha helix or beta sheet. If both the proteins are given, then there will be some changes. So although secondary structure analysis can be done, but the high resolution 3D dimensional structure cannot be determined using the circular dichroism. Sorry. Next question is CD spectroscopy relies on the measurement of. Hmm. So CD spectroscopy, it it is the first option. It will be absorption of difference of left and right circularly polarized light. That will be the basics for the measurement of the CT spectra. Okay, all the options are incorrect. The first option is correct here in this case. Then we have this structural and functional unit of protein which functions independently. Okay, so yeah, we have folds, turns, domains and motifs. Out of these four, which is the structure and functional unit of the protein and has the capability to function independently? Is fold the correct option? What are folds, what are turns, what are domains and motifs? Okay, so in the secondary structure, okay, we have this folds. Okay, and from one fold or from one structure to another, we there is a loop or turn. Okay, and then another structure starts. So these kind of turns are present due to presence of glycine or proline in, so that generally discontinues the secondary structure and makes a turn there. Okay, so but these folds and turns are not any independent functional units. Okay, then comes domains and motifs. Okay, so motif is you can say um, series of secondary structure. You can say a super secondary structure. Okay, so motifs are referred as super secondary structure where will be uh, a series of secondary structure you can see. Okay, so for example, I have motif as you can say like hairpin motif or triple helix motif, or you have helix turn helix motif. So there are different kinds of motif. Okay, we have helix turn helix. Okay, so this is one helix, then we have one turn, and then again one more helix. So this entire thing is referred as motif. Okay, so generally helix turn helix motif is known to interact with the DNA. It is a DNA, it is having DNA binding property, but it is not a functional unit. Okay, however, domain is a in 3D dimensional, it shows a particular portion of protein which is able to give functionality to the protein. Okay, so domains are decided three dimensionally. However, motifs are some sort of super secondary structure that are present in the protein. So domains can function independently. Suppose you have one such big protein and you can take one domain. That domain has some particular activity which can be performed independently. Like you have a full length protein known to perform in certain pathway. Okay, but if you only take one domain of that protein, suppose it has ATP hydrolysis activity, so if you just take one domain, okay, remove make a truncation of only one domain, it will still be able to perform that helicase activity because that domain is providing that protein that activity. So that is the difference. Motifs of the supersecondary structure cannot function independently. Okay, and domains are such three-dimensional structure which has the capability to function independently. So the correct option will be C. Okay, this refers to the unique three-dimensional conformation that globular protein assumes as a consequence of the interaction between the side chain. Okay, so what will be the correct option? It's very easy. 3D structure. Okay, 3D structure. Does primary and secondary structure will give you the 3D structure? Primary and secondary structure are also formed between the because of the interaction between the CO and NH group. Okay, not because of the side chain interaction between the side chain, right? So the correct option here will be
C.
sorry. Please say it again.
Tertiary structure.
Yes, the correct option will be the tertiary structure. Yeah. So dash is super secondary structure which is formed by connected alpha helices and beta pleated sheet through loops, exhibit similar function through protein. Yeah, so as I said, this helix turn helix, if this structure is present in any of the protein that has some particular function but cannot function independently, so that is something which is motif for you. Okay, so this HTH motif known to bind DNA. Okay, as it has a turn of approximately 4 amino acids, so those are some structural uh, requirement. Okay, and helix loop helix, so this is turn, this is making a loop. Okay, DNA binding motif, and then coiled coil. So these are different types of motif. So if it is a super secondary structure connected by alpha helices beta pleated sheet through loops turns, it is nothing but motifs, right? Yeah, then we have this question which is homology modeling relies on the fact that homology modeling, what is homology modeling? homology modeling refers to the, like suppose you have a protein, okay, and you know the function and everything, okay, you have the structure of the protein. Now you find the similar protein in another organism, okay. So because based on that structure, based on that sequence of the protein, you make a homology model that is matching with the known protein, okay. So that is homology modeling. So that is based on what fact it has been asked. Please try to answer. So first option is amino acid sequence determines the tertiary structure of the protein, okay. So uh, so based on the sequence only, so I have one protein known to me and I have the 3D structure of that. I have another protein which has, I know the sequence, okay, and based on the sequence only, I will make another model and then try to superimpose both the models with each other and see whether it has a similar kind of loops or domains which are found, okay. So that is called homology modeling. So that is based on the fact that the amino acid sequence determines the tertiary structure of the protein, because whatever the sequence will be, the same kind of protein polypeptide it will form, and because the polypeptide have will having similar interaction in the two secondary, as a secondary structure and tertiary structure, it will, the overall structure of the protein will be the same. So the fact is the amino acid sequence determines the tertiary structure of the protein. The correct will be first, okay. All others are not correct, simply. Okay, so this is interesting. In an experiment, gel filtration of protein sample deduced molecular weight of 54,000 Dalton, whereas the same protein was run on SDS page and a thick band corresponding to 18,000 Dalton was observed. Based on the observation, the protein is, okay. So how many of you knows about these technique? What are these technique? What is gel filtration? Anyone try to answer? What is gel filtration? You all know the protein purification techniques. Like you are working on a protein, you are interested to work on a protein, and you like express this protein in some another organism, for example bacteria. So along with your protein, all the bacterial proteins will also express, right? So there are several techniques to purify your particular protein of interest, okay. Among that it comes gel filtration, okay. So you have performed the gel filtration of the protein sample. So that is my question. Can you answer what is gel filtration? Anybody, please unmute yourself. Okay, so gel filtration is nothing but uh, sorting the protein based on its size. Okay, we have a column, okay, filled with such resins which are porous in nature. Okay, then we have the proteins. Okay, some smaller proteins, some bigger proteins. Okay, so the smaller protein will go through the pores to pass this column, but the bigger protein will just easily can pass the column. Okay, so as the smaller protein is taking time to pass through the pores, the longer or the bigger protein will come out first from the column. So first this bigger protein will come, and next second the smaller protein will come. Okay, so based on that, when you'll elute, you'll have a bigger protein sample. So from the gel filtration, the protein sample deduced molecular weight of 54 kilodalton. So what they have got, one protein which has the molecular weight of 54 kilodalton. Okay, so this is the molecular weight they, or this is the protein of 54 kilodalton they got from the gel filtration. Now they run the protein on SDS page. What is SDS page? You people know? What is SDS? Please try to answer. Please interact.
Polyacrylamide gel.
Yeah, and SDS is,
Sulfur diester.
it is a detergent, sodium dodecyl sulfate, right?
So
Yeah, so what does it do? You are giving a detergent, a harsh reagent to protein, so it will break all the bonds. Okay, it will bring the protein in its tertiary, from its tertiary or quaternary structure to its primary structure, right? It will basically, it will denature the protein by breaking the interactions among the protein or among the amino acids. Okay, so SDS makes the protein, brings the protein at its primary structure. Okay, so here the protein you have got is 54 kilodalton. Then you run it on the SDS page, and on SDS page what you are getting, you are getting a thick band corresponding to 18 kilodalton. Okay, only one band. But when you eluted the protein, its size was 80 54 kilodalton. So it should come above, near, like at higher weight, but it came at the lower weight. Okay, because SDS denatures the protein, breaks all the interactions which are present in the protein. That means the protein has some interaction among the residues, and that is why it was giving a high molecular weight. Okay, so the protein has more than one polypeptide unit. Okay, so now you have to find out whether that uh, all the different units of the protein are of the same polypeptide or from different. Okay, so you have homodimer. First option is homodimer. Homo means there will be the same polypeptide, and dimer, two forming together one particular protein. So if 18 into 18, to 18 kilodalton polypeptide altogether, then it will make a 36 kilodalton protein, right? But you got 54 kilodalton. So it is not homodimer. Then it has been asked, it is heterodimer. Okay, so if it is a heterodimer, it will be, the polypeptides will be of different size, but you are getting only one band of 18 kilodalton. That means all the polypeptides are of same size, because of that only it is coming on the same size. So it is not heterodimer, it is of the same size only. This is also wrong. Now it has homotrimer. Okay, so similar polypeptide. Okay, so each of 18 kilodalton. Okay, altogether this is making a 54 kilodalton protein. Okay, so it will be homotrimer. Okay, and because in the gel extraction you are eluting the protein in its native form, that is why it is coming as 54 kilodalton. So this is the quaternary structure of the protein. Okay, and because more than one unit is being joined, so it is quaternary structure. And when it is run on the STS page, which is has denatured all the function interaction, so all three 18 kilodalton subunits are separated from each other, and because they have the similar size, when they are running on the STS page, they are coming on the same size. Okay, so that is the reason you are getting a 18 kilodalton band. Is it clear? I hope it's clear. No one is answering, no one is saying anything. Okay, so these were the questions. You have any doubt you want to ask me? Okay, if no doubts, I would like to conclude today's session. Thank you.